In this video, we'll give you a quick overview of the different online tools you can use to design guide RNAs for your CRISPR experiment. If you would like a guide RNA for your CRISPR knockout experiment, choose CRISPR Knockout in the first drop-down menu. Next, enter the name of your species or organism in the text box and select it below. Then, enter your gene target by typing the gene name or gene ID. You will have the option to purchase pre-designed guide RNA to your gene of interest in multiple product formats, including synthetic single guide RNA, synthetic CRISPR RNA, as well as all-in-one CRISPR-Cas9 lentiviral single guide RNA, which combines CRISPR single guide RNA and Cas9 nuclease expression into a single lentiviral package vector for the simplest gene knockout and workflow available. You may also purchase lentiviral single guide RNA to be used with a stable Cas9 cell line. If you would like to design guide RNAs for a specific region of your gene, for example, if you are conducting a homology-directed repair experiment, choose CRISPR site-specific editing in the drop-down menu. Guide RNA designs near the location of the desired edit are available as synthetic single guide RNA, synthetic CRISPR RNA, all-in-one lentiviral single guide RNA, and lentiviral single guide RNA. If you already know the guide sequence or sequences you would like to order, click on Order Guide RNA. The sequences can be added individually or copied from an Excel file. Your guide RNA can be ordered as synthetic single guide RNA, synthetic CRISPR RNA, all-in-one lentiviral single guide RNA, or lentiviral single guide RNA. If you know the DNA sequence where you would like to make an edit, choose Design Guide RNA. Once you have chosen your target sequence, your guide RNA can be ordered as synthetic single guide RNA, synthetic CRISPR RNA, all-in-one lentiviral single guide RNA, or lentiviral single guide RNA. We hope you enjoyed this video on how to design custom guide RNAs for your CRISPR experiment. See you next time!